Ford asked me to make a few comments about the remarkable process by which a vaccine moves through the regulatory system. That is, from a beginning idea, such as we've heard from several speakers, Barney Graham, David Leap, and others, Jason McClellan, and how it then becomes a commercially viable vaccine that's licensed by the FDA, and then guidance is issued by the CDC, and then finally recommendations are made by the individual states as to what sort of vaccine requirements will be applied. So the vaccine story usually begins as a conception in the mind of creative individuals, or perhaps the sponsor may be a small startup biotechnology company, or it may be a large pharmaceutical company. But the first step is to submit an IND, which is an investigational new drug, to the FDA. That is performed in order to ensure that the rights and the safety of the participants in the trial will be carefully followed. And it's also a review by the FDA to ensure that the study will be conducted with scientific rigor and produce results that can be interpreted. 30 days after the IND is submitted, if there has been only a reply or acknowledgement of receipt, or there is no response from the FDA, then the sponsor can begin the application for a BLA, which is a biologic license application. BLA is essential in order to introduce a vaccine into interstate commerce. So the requirements must be carefully fulfilled. The clinical aspect begins with a phase 1 trial, which is typically fewer than 50 subjects, otherwise healthy subjects who were closely monitored during the period of observation. And the primary interest during the primary phase, or the phase 1 trial, is to determine safety. If events go well, then a phase 2 is begun. Phase 2 is more focused on efficacy. This involves typically a few hundred subjects, and individuals are followed, of course, for safety, but also for evidence of efficacy. And if either one of those thresholds is not met, then usually development of the vaccine is discontinued. But if it's successful, it goes on to phase 3. And the third phase is traditionally the randomized placebo-controlled trial. If there's a standard of care, the standard of care is used in the non-experimental vaccine arm. If there is no standard of care, as with the COVID-19 vaccines, then a placebo, an inactive placebo, is used as the comparator. And this phase 3 trial typically consists of thousands, maybe 10,000 subjects, depending on the vaccine. With the messenger RNA vaccines that we've just been hearing about, these trials involved as many as over 40,000 individuals who were randomized either to a placebo arm or to receive the experimental vaccine. There is a phase 4 trial that is conducted sometimes after a vaccine is licensed or authorized, and after it's been widely available, and this is to look for specific side effects that were not picked up in the phase 3 trial, such as the rates of myocarditis that were not detected until phase 4 trials were really initiated. The route to completing the FDA is very complicated. The FDA, of course, looks at the primary data that was collected during the phase 3 trials. The FDA does their own biostatistical analysis. They visit the clinical sites, trial sites where the vaccine was studied. They look carefully at the manufacturing process. They go to the plant where the vaccine is indeed made and look at all of the supply components that go into the vaccine to ensure that it all meets the requirements for appropriate preparation. That's the standard route by which a vaccine becomes licensed. There are alternative routes. There is the emergency use authorization, which we know about so recently after the COVID-19 vaccines. And there is the expedited access or compassionate use vaccine trial that was used recently in the last 10 years when there were outbreaks of meningococcal B disease on a number of campuses around the United States, particularly at Princeton. And this gives people access to a likely intervention that has not yet been fully authorized. There's also the animal rule. For example, the anthrax vaccine was licensed because it's not practical to conduct that trial in humans. So during the development of the SARS-CoV-2 vaccines, the FDA called upon an advisory committee, VRBPAC, which is the Vaccines and Related Biologic Products Advisory Committee. And we met, actually, counted up the number of meetings. There were 17 meetings between June of 2020 and July of 2023. And that committee was was chaired by Arnold Monto, who did a remarkable job. After approval or licensure or authorization is issued by the FDA, the next stage is taken over by the CDC. The Advisory Committee on Immunization Practices makes a recommendation, and the ACIP votes on how the vaccine might be used. It is then up to the individual states to decide how they want to use that guidance that then comes from the CDC. So I'm going to stop at that point, and shall I start the first question? Yeah. Okay.
Cody, excuse me, did you want to tell your story about Sam Katz?
Oh, I would love to. Thanks. But I didn't want to use up too much time, so thank you. Yeah, and I actually haven't told this yet to Ford and to Peter, but my first encounter with Sam Katz was just after I finished my infectious disease fellowship with several of the people here in the room. And I wrote a paper about the pandemic potential or the epidemic potential from influenza virus. And in that article, I misspelled one word. And the next day, or a day or two later, I received a note from Sam, and he said, Cody, I very much enjoyed reading your paper, but you misspelled waterfowl. I had spelled it F-O-U-L. And despite endless readings all of us who publish papers know that occasionally something slips by. And I wrote a letter back to him and I said, "Dear Dr. Katz," because I was thrilled that a man of his stature, had read the paper, and told him that I would do my very best not to make an error in spelling in the future. And then a few years later, I published an editorial in JAMA making the point that I didn't think we were appropriately evaluating the importance of vaccines to society. And 2 days after that was published, I received a message from Sam Katz, and he wrote, "Dear Cody, we should all thank you for writing such a wonderful paper, Sam." And to me, it exemplified his mentorship, which Peter had mentioned earlier. He really was concerned about the development of younger research people who are interested in vaccine research. So it meant a great deal to me. Okay, so I will begin with the vaccines. Questions, and I've got one question. David, you're first up, and the question is, okay, thanks. Have you looked at how your AAV, adenovirus-associated virus system, might help with viral clearance of reactivated HPV in patients where it has been latent?
So I'm assuming it's HSV, not HPV.
It says HPV, but I'm assuming.
Well, we've never looked at HPV. I can answer that one very quickly.
Okay. I think you're right.
But we are in the process right now of collaborating with a group, David Bernstein's group in Cincinnati, to look to see whether or not these AAV vectors can express antibodies to modulate reactivation in a guinea pig genital model. So one of the disadvantages of the mouse system is that you don't see robust in vivo reactivation. And so we've switched to a different animal system with a collaborator to investigate that. And we're anxiously awaiting beginning those experiments.
Okay. Liz, the next question is.
We should alternate.
Oh, okay, sorry.
Yeah, no, that's.
Sorry. Get my stuff out of here.
All right, so Dr. Graham, I'm going to pose this one to you from the audience. How can we potentially combat fear of potential long-term side effects for the RSV vaccine? No pressure.
Can you use the microphone?
Oh, yeah, sorry.
Yeah, the question is, how do we combat or push back against fear as we start the RSV vaccine program? And one of the reasons I put that link up there from the work the journalist did on the vaccine enhanced illness story is that, RSV is primed to have anti-vax sentiment, and there's a lot of information out there that could be turned into anti-vax sentiment. So my hope is that these vaccines have very few side effects, and so my hope is that they're well tolerated and that we, see that they're working and somehow communicate that in a way that people can hear it and we don't have to go down that path. But I've been worried about just that problem. I don't know the solution for how to, the only way you can get trust is to be trustworthy, and it takes time to be trustworthy and demonstrate that. So I don't know that there's a way that I know of to really combat that other than being as trustworthy as possible and transparent as possible. That's why I'd like this story to come out sooner rather than later.
Barney, Dr. Graham, why do other delivery methods for children, why would they be more likely to avoid the prefusion? Yeah.
Yeah, the question is about, why did I list those gene-based delivery approaches for the vaccines in the 6-month-old? And so what was learned back in the 1960s is that if children were already infected with RSV, or if they even got live attenuated virus as an IM delivery, that they did not get the vaccine enhanced illness. And so it was really whatever happened during that priming event in antigen naive children that set up the immune response to either have bad T cells or bad B cells. And so now that we have a well-designed vaccine, a well-designed antigen that creates high-quality neutralizing responses, maybe we've solved the problem. And maybe that protein could be safe in young infants. But if there's a T cell component to that, the way to avoid the Th2 responses is to have an early CD8 T cell response, because in settings where you prime the immune system with a CD8 T cell, it makes so much interferon gamma that it forces all the CD4 T cells to differentiate the right way. And so that's why most of us who've been doing this for so long think that those gene-based delivery approaches that can induce CD8 T cells are likely to be safe as long as they are using good antigens.
Dr. Brickley, this one's for you. Can you tell us a little bit about continued challenges in polio that remain on your mind?
Thank you so much. Well, I think the major one is that polio remains, as of May this year, it's our only public health emergency of international concern. And the reason for that, so in that map I showed you, we had 10 cases of wild poliovirus, cases of polio last year. But all of those other dots, the more than 300, were from something called circulating vaccine-derived poliovirus. And one of the major challenges is so circulating vaccine-derived polioviruses can emerge in under-immunized populations, and they come from our oral live attenuated polio vaccines, which in these under-immunized populations, whenever they start to circulate, they can reacquire some of that neurovirulence and actually cause polio themselves. And so the big challenge is that our best tool for fighting these circulating vaccine-derived poliovirus outbreaks is to use the oral polio vaccines that led to them, and so you can seed further outbreaks. But I think this is also something I'm most excited about is so we have these novel oral polio vaccines that have been engineered to be more genetically stable. And so far we've been using type 2, and really the evidence to date, so it started to be used in March 2021 under an emergency use listing. And we've since then administered more than 800 million doses of it. And what's really encouraging is that we've seen that so far it appears to be very safe. Our research together with Peter has shown that it's inducing really kind of equivalent mucosal immune responses to the traditional Sabin OPV. And what we're also seeing that's really exciting is that we're not seeing the emergence of new outbreaks of circulating vaccine-derived polioviruses to the same extent. So the estimate is we've seen something like 3 emergences from the novel oral polio vaccine, versus during that same time we would have expected to see maybe 40 emergences from the traditional Sabin OPV. So the evidence to date on the novel OPV type 2 is really encouraging, and so it'll be really exciting to look to the future and also to think about the other serotypes, but also thinking about how these can potentially be a tool as we see the declining population level immunity due to gaps in vaccine coverage, that OPV is not completely out of the story yet. There's a lot of reason for hope.
Margie, next question is for you. Can you comment on the future role of decentralized clinical trials?
It's a great question. I mean, I think we're seeing beyond biomedicine, right, huge efforts in data aggregation. And we worry about that in some spheres, right? Losing privacy, losing, having outcomes that come from aggregation of data that are unanticipated. But I think that's one opportunity that's a bright side from those efforts. And so more passive surveillance too may be possible to gain further insights that come from those efforts. I think you'd probably have to ask a clinician for more insights kind of into how that might work, or you could answer operationally from the regulatory perspective, what the barriers and the value proposition might be. But I think on the data end of things, decentralization and certainly distributed deployment of trials has a lot of value and appeal.
Thank you.
Okay, this is a tough one. Dr. Graham, I'm going to give it to you since you've worked at the institutional level quite a bit. How can we change the perception of vaccine development as unprofitable outside the pressures of a pandemic? And if you want to take a pass.
Say that. Just say that one more time.
Right, sure. So how can we change the perception of vaccine development as unprofitable outside the pressures of a pandemic?
Yeah, this is a hard one because it is, as we try to be better prepared for the next pandemic, a lot of what I think needs to happen is distributive research and distributive manufacturing. And so you can start solving regional diseases by regional investigators. And so I always pick on Rift Valley fever. So, I think the Kenyans need to solve Rift Valley fever. And the problem is, and I think mRNA gives us a chance to do that sort of thing, but the problem is if there's a commercial mRNA vaccine, for instance, for Rift Valley fever, that market may only be 100,000 to 1 million people a year. And that's not a big enough market for most for-profit pharmaceutical companies to pay attention to. So one of the things I think we have to do is find business solutions to make small market vaccines feasible and viable. So you have these sites that can be surge capacity during pandemics, and we can solve the regional problems before they become global problems.
Well, thanks everybody for your comments. I want to ask one last question. And I'd like to ask Dr. Graham if you could kind of look into your crystal ball and see which pathogens are you're most concerned about in the future, and what would you suggest that we do to prepare for the next event?
Okay, well, I'm glad you asked that because I don't believe in prioritization. I think we have the tools and the capacity to do all of it. And so I am helping the WHO to lead an effort right now where we've collected 26 committees to look at the 26 viral families that infect humans and have those 10 or 12 experts on each of those committees have a discussion about, what are the viruses of greatest concern within each of those families. And then I'm the chairman of the chairman, and we get together and we talk about, what should the priorities be. But I think all the viral families need a similar solution using prototype approaches that can be generalized like we were able to do with coronavirus, and that combining that with rapid manufacturing like you can do with nucleic acid vaccines can provide answers. And that if we prioritize, it should be the priorities of the actions within each of those viral families. Some need better surveillance, some need better vaccine solutions, some need immunology breakthroughs, and each of the viral families really has a different set of issues that I think those action steps could be prioritized. But putting one above the other and just working on the top of the list, I think, is self-defeating, in my opinion.
Interesting comment. I think one of the lessons that we've learned from the current pandemic is the importance of a joint effort by the federal government, by the pharmaceutical industry, and by individual sites such as academia. And I think that continuing to work together will offer the greatest promise for the next pathogen. So thank you very much.